small amount of concentrated urine. When you're down the pub drinking tons of beer and you're not sweating, then you produce large amounts of very dilute urine. How does that work? It's very difficult for the kidney to do it, but it does it, and very efficiently, as you know. So we have to deal with this question of how we vary the concentration of our urine depending on how much water you want to keep. If you're dehydrated, you want more, and how much you can afford to pee out if you're not dehydrated. So that's the most important thing today. So bear with me while I flip through some of these early slides on sodium and potassium, which I wanted to show you, and you can go through at your leisure, but I'm not going to spend a lot of time on them. Okay, good to go? Can I take that off? Otherwise you'll be worrying about it all through the class. Alright, so sodium is transported by active transport. I'll show you pictures of this in a minute. Chloride is transported passively, following the imbalance caused when sodium is pumped out of the cells. Potassium, now sodium, when we talk about sodium, we largely talk about reabsorption. In other words, you're concerned with not losing sodium. You're concerned with reabsorbing sodium into the blood and not losing it in the urine. Potassium is a little bit more complicated, as I'll show you, because depending on the region of the nephron, potassium can be either reabsorbed or secreted. Remember, secreted means you lose it in the pee. Reabsorbed means you keep it in the body. So we have mechanisms very carefully for controlling potassium, and the reason for that is that there's a lot of potassium inside the cells, and potassium is essential for electrical activity of things like the heart and muscles and electrically excitable tissues. So potassium is complex for that reason. So here's this is a proximal tubule cell. Here's the route of reabsorption, quite straightforward. Here is the tubular lumen, here is the filtrate. Here are the tubular epithelial cells connected by tight junctions. The route of reabsorption is from the filtrate into the tubular epithelial cells, from there into the interstitial fluid surrounding the tubule, and from there into the plasma. So that's reabsorption, and that's the pathway that sodium ions typically will take. And here is just a rough idea of which region of the nephron handles sodium in different amounts. And as we talked about last time, reabsorption, you think reabsorption, you think proximal convoluted tubule. So about 67% of sodium is reabsorbed in the proximal convoluted tubule, some in the thick ascending membrane. We're going to talk about the leukopendy today, and then a little bit in the distal tubule and collecting duct. And you excrete less than 1% of what you reabsorb. So that's reabsorption of sodium, largely in the proximal tubule. So as we looked at last time, you remember that glucose picture we finished with last time? So that's the kind of picture we're going to look at for the next few minutes. And you'll remember how that works. The sodium diffuses into the cell at the luminal membrane, and it's pumped out of the cell by active transport in the proximal tubules at the basolateral membrane. Remember, that was the sodium-potassium pump going on at the basolateral membrane, and it's diffusing in on the luminal membrane. And that diffusing in, there are various transporters there, and one of them, you remember, was the glucose-linked cotransporter. So we talked about that last time, and I'll show you again in a minute. And the active transports, you know about this, sets up an electrical potential difference. You know this issue of minus 70 millivolts inside the cell, which also pulls in sodium from the lumen. So sodium is responding to a concentration gradient and an electrochemical gradient. So here it is. So here is here's the lumen, the tubule cell, the interstitial fluid, And here is the basolateral membrane with the sodium-potassium pump going on, pumping sodium out of the cell into the interstitial fluid. See the size of the Na, small there, big there, and that allows sodium to diffuse into the cell from the lumen. And this is there are several sodium channels here, and one of them, as you remember from last time, is linked to glucose. We're not going to put glucose or anything else on here right now. So this pumping out then causes a minus 70 millivolt potential difference. The lumen is about minus 40, 0 inside the cells. So the sodium then is going from the lumen to the cell, pumped out here and diffuses into the blood. So that is the route of sodium reabsorption, active transport at the basolateral membrane, and diffusion at the luminal membrane. We talked about that last time, essentially. So two factors move sodium through the brush border: the concentration difference, small sodium, big sodium, and the electrical potential difference, 40, 70, 0. Movement of chloride is passive, and water passes from the lumen through the cell into the interstitial fluid by osmosis, which we talked about. And as we said before, 99% of the water that you filter, you reabsorb. Remember, filter about 125, pee out about 1. So 99% of the water in the filtrate is reabsorbed, and it's reabsorbed largely in the proximal tubule cell again. So here is the same picture as last time, with water added to it. So here is our sodium-potassium pump at the basolateral membrane. So sodium is being pumped out, potassium in. That allows sodium to diffuse in, down its gradient, and then into the tubular capillaries as last time. But look what the effect of that is. The effect of that is to lose sodium from the lumen, and you have a local decrease in osmolarity, allowing water to diffuse in through the cells and to some extent between the cells to where the local osmolarity for sodium is high. So that is how water follows sodium by osmosis, because the osmolarity changes there due to the pumping of sodium unidirectionally across here, causes local osmolarity changes, which causes water to move in response to the osmotic gradient set up by the sodium ions. So water, all this says is that water follows sodium. Now this is, and the previous one, is really the proximal tubule cell, although it doesn't say so. But here is the cortical collecting duct. So put a ring around this, because it's easy to miss that. This now is taking place in the collecting ducts, the distal tubule and collecting ducts. There's a gradient between the distal tubule and the collecting duct. There's overlapping functions between these two. This says collecting duct. You'll see on the next slide it says distal tubule. That's because there's overlapping functions between these two regions. So here we are now in the collecting duct region. And what is going on? Same thing here. Look, sodium and potassium pump at the basolateral membrane. Sodium high on the interstitial fluid. Potassium high in the cytosol. And look, the cortical collecting duct cells have potassium channels. These weren't shown in the proximal tubule cells because they're much more important here. You remember I said that potassium can be handled both by reabsorption and secretion. And so the secretion is happening in the collecting duct and distal tubule because these cells have potassium channels here. So the potassium then is pumped in, it's high here, and because there is a channel here to allow it, potassium diffuses in to the lumen through the potassium channels. So this pathway here, small potassium, big potassium, small potassium, is potassium secretion going on in the collecting duct and distal tubule. So potassium is more complicated. Here it is. It's removed from the filtrate by reabsorption in the proximal tubules. Think proximal tubules, think reabsorption. But it's secreted into the distal tubule. See, this is distal tubule and collecting duct by active transport from the peritubular fluid. And it's important that you have this balance between reabsorption and secretion because levels of potassium are important in the body. So potassium balance is essential for normal function of excitable tissues. 98% of the body potassium is intracellular. You remember way back last week, one of those very first slides I showed you between the difference between intracellular and extracellular fluid? Potassium is all intracellular, mostly. So that's why potassium is important. And so here is an equivalent picture to the one I showed you for sodium for potassium. And so here you are, proximal tubule, reabsorption. There you go, 67%. 20% in the ascending limb of the loop of Henle. This is called the descending limb and the ascending limb. And here, there is a variable secretion. So this is where that secretion is happening. In the distal tubule and early collecting duct, there is variable secretion of potassium depending on a variety of things. Your dietary potassium, a hormone called aldosterone, which I'll talk about, acid-base balance, and flow rate. So I'm not going to go into this in detail. It gets very complex, but it's important, and potassium levels are very important. You go to the doctor, he finds you have hypertension, high blood pressure. The first drug, if he gives you a drug, or she gives you a drug at all, will probably be a diuretic. We'll talk about diuretics next time, or on Monday. Diuretics pee out water and lowers your blood pressure by peeing out water. The problem with that is that you might pee out too much potassium. So you go to the doctor, he or she gives you a diuretic, you go back in 6 weeks, brilliant, your blood pressure's down, we need to check how your potassium is doing. Because if you pee a lot, you're likely to lose potassium, and that's bad news. And so if the doctor finds that in fact you are peeing out potassium, then they have to adjust the diuretic that they give you. Some of them are better than others than keeping potassium in the body or losing it from the body. So it's an issue, and it's a complex issue, and I'm not going to go into it in detail here, but the way the kidney handles potassium is an issue, but it's by balance of reabsorption and secretion. And so look, you excrete between 1% and 110%. What the heck does that mean? You excrete more than you filter? I don't understand that. It's variable depending on conditions in the body. So that's the bottom line there. Sodium, mostly reabsorbed in the proximal tubule. Potassium, combination of reabsorption in the proximal tubule and secretion in the distal tubule collecting duct. There you go. Potassium is handled in the kidney by a combination of filtration, reabsorption, and secretion. Distal tubules and collecting ducts adjust for dietary intake by varying the rate of reabsorption and secretion. Most excreted potassium is the stuff that's secreted from the collecting duct. What's next? Let me see. Okay, so that's what I wanted to say about sodium and potassium. Fairly straightforward stuff. You can read through it. I know I went through that fast, but I think you can handle it. What I want to talk about now is water. And it flows naturally from what I've just been talking about. Because, again, 67%, I don't know where they get this figure, 67%, it's very exact, isn't it? 67% of filtered water is reabsorbed in the proximal tubules. As I showed you, the driving force is the osmotic gradient across the tubules set up by solute reabsorption, mostly sodium. Remember I showed you that just now in that other picture. and therefore changes in sodium reabsorption alter water reabsorption. So you eat a full big bag of salt and vinegar potato chips, you take in a lot of sodium, you pee it out, and you pee out a lot of water with it. So dietary changes affect water excretion, but mostly it's a state of hydration. So here is a similar picture for water reabsorption. Proximal tubule, yeah, reabsorption, there it goes. 67%, I don't know, 70% rounded up, most of it. Thick ascending limb, 20%. We'll talk about that in a minute. Cortical collecting duct, 5%. And you pee out 1%. Now, this is a general picture, and I don't want you to remember these numbers particularly. What I want you to remember from this picture is introduction to the fact that your peeing out of water is dependent on the collecting ducts under the influence of this substance called ADH. Okay, we're leading into this issue now of how you adjust the concentration of your urine depending on the state of hydration. It has to do with water reabsorption in the collecting duct under the influence of ADH. Do you know what ADH is? Antidiuretic hormone. Now then, for the next couple of classes, I'm going to be talking about hormones. Sometimes we organize these lectures and endocrinology comes before kidney in the lecture schedule. That's nice because you know all about antidiuretic hormone and you know all about aldosterone that I have to talk about. But you haven't done it, so I can't assume that you know about it. So I'm going to have to talk about these hormones. And then when you come back and do endocrinology toward the end of the term, you'll do it again with the focus on the endocrine aspects. Here we talk about the kidney aspects of it. You know, it doesn't matter which order we put these lectures in. If you do endocrinology first, that's fine. The endocrinology lecturer will tell you about ADH and aldosterone and its effects on the kidney, but you haven't done the kidney, so you haven't got a clue what he's talking about. And if we do it this way around, I have to talk about ADH and aldosterone, and if you haven't done endocrinology, so you haven't got a clue what I'm talking about. So either way, you have to put it together at the end. So I'm going to talk about these hormones as if you don't know what they are, which is probably fairly accurate. And then you will do them again when you talk about endocrinology. Yeah? Anyway, so the hormone we have to talk about here, the kidney, by the way, is the target of quite a number of hormones. And I'm going to talk about those specifically next time. But the one we're most concerned about today is ADH. And ADH is a hormone called antidiuretic hormone. Hey, that's cool. Antidiuretic hormone. Diuresis is peeing out water. So antidiuretic hormone is a hormone that prevents you peeing out water. Very important. ADH. Now, it's sometimes called vasopressin. And when you come to do endocrinology, you'll find out why. But if you read about vasopressin, it's the same thing as ADH. I'm going to use the term ADH. In some textbooks or some classes, you will hear the term vasopressin. I know that's confusing, but that's how it is. And there's a reason for it, but I'm not going to go into that today. It'll come clear later. So, ADH or vasopressin directly regulates water reabsorption in the collecting ducts. This is the key to understanding this part of the kidney. Other hormones also regulate water reabsorption indirectly as a result of their effect on sodium reabsorption. So, because sodium and water are linked, if you change sodium, you change water. And there is a hormone that does change sodium reabsorption, which we'll talk about, called aldosterone. But I want to put that on one side for a minute and focus on the effects of ADH first, or vasopressin. And what happens is, and we'll have a look at this in detail, the water passes through special channels in the membranes of the cells of the collecting duct called aquaporins, usually abbreviated AQP. And this is very sexy modern recent research. And the aquaporins are always open in the proximal tubule cells, so that's why the proximal tubule cells are able to reabsorb lots of water all the time. But in the collecting ducts, they can open and close. So if you can open and close the water channels, you can affect how much water you pee out and how much water you keep in. I'm going to explain that in detail. So it all has to do with these water channels in the collecting ducts, which are under the influence of ADH. So here's the main part of today. Gets tricky. I need your concentration for about 20 minutes. You think you can manage that? Okay. So we've got all this stuff going on, but it has to be responsive to the needs of the body for water. If you're dehydrated, you can't be peeing out water, otherwise you become more dehydrated. So you conserve water. And so the kidneys save water for you when they produce hyperosmotic urine. In other words, concentrated urine, which is more concentrated than the body fluids. Remember what the body fluid concentration is, roughly? 300 milliosmoles. I asked you to remember that number. 300 milliosmoles. We're producing urine. When you are dehydrated, you're producing small amounts of very concentrated urine, much more than 300 milliosmoles. How the hell do we do that? Okay. It's carried out by a mechanism called a countercurrent mechanism. Now, I know that you're not engineers. Are there any engineers here? Not out of the question. Some engineers want to do biomedical engineering, so they come from the engineering faculty into a class like this. And there aren't any today. But if there were, engineers would know about countercurrent mechanisms because it's an engineering concept. It's an engineering concept because a countercurrent mechanism is a multiplier. And the engineers use it for heating purposes. And so what you do is you have tubes going through a system like this with flow like that in one direction in one tube and flow like that in the other direction in the other tube. And the interaction between these flows multiplies heat in the system. Now, we're not multiplying heat in the system. What we're multiplying is sodium concentration. So a countercurrent multiplier, you could put a countercurrent multiplier there, enables you to concentrate sodium. Why is this important? Well, it depends on this anatomical arrangement of the loop of Henle and the vasorector. Finally, we're going to talk about what it is that these long loops of Henle do. The long loops of Henle that go in the renal medulla. Now, have you noticed on these pictures that I've shown you, everyone, right from day one of kidney classes, have you noticed that the background here in the medulla is always a different color from the background color in the cortex? That is meant to illustrate the fact that the extracellular fluid in here is much more concentrated in sodium chloride than out here. So that's what that gradient, there's a gradient in the kidney from the cortex to the medulla of increasing sodium chloride concentration. Ooh, haven't mentioned that before. So, and this is called the osmotic gradient in the renal medulla. So out here, the sodium chloride concentration is relatively low, and it gets more and more as you get down here, and it's very high down here. Now, we're not talking at the moment about what's in the tubule. Got to get this in your head. We're not talking about what's in the tubule. We're talking about what's outside. So out here. Right? And this gradient is the key to how we produce concentrated or dilute urine. So we have to understand how the gradient is set up. So I'm not going to talk about what's going on in the tubule for a minute. I'm going to talk about how the gradient is set up in the cortex and the medulla. And that is the job of what's going on in the loop of Henle. See, we've got a countercurrent going on here. We've got a long tube going down, and right next to it we have a long tube coming back up. We've got this countercurrent multiplier going on. So let's have a look at how the loop of Henle sets up this gradient in the extracellular fluid. So the osmolarity of the interstitial fluid in the cortex is about 300. So you can write 300 milliosmoles here. And down here, in the bottom of the medulla, it's about 1400. Way more concentrated. And the osmotic gradient is produced by a countercurrent multiplier, which is the loop of Henle. So let's see what's going on in the loop of Henle, how it sets up the gradient, and then we'll see why that's important for producing different concentrations of urine. And that has to do with ADH. So we'll come back to that. Okay, here you go. So this countercurrent mechanism occurs both in the loop of Henle and in the vasa recta. You remember the long, straight blood vessels that surround the loop of Henle. And it has to do with the differing permeability of the cells on the way down and on the way up. Remember I said that the nephron is brilliant because it has all these different segments, and all the different segments have different permeabilities? Well, the difference in permeabilities in the loop of Henle are vital for the setting up of this gradient. I'll show you a picture of this in a minute. This is just the statements. The descending limb of the loop is highly permeable to sodium and chloride. The ascending limb has a strong active transport pump to remove sodium from the tubule and is impermeable to water. Water does not leave the ascending limb, so the concentration decreases. So let's see what that means on this picture here. So here is the loop of Henle. There's a thick descending part, and a thin descending part, and a thin ascending part, and a thick ascending part. All have different functions. The thick descending limb is basically a straight proximal tubule. We don't have to worry too much about that. The thin descending limb has low passive permeability to ions, but high permeability to water. I hope this will make sense in a minute. I'm just showing you what the situation is, then we'll see why this is important. The ascending limb, the thin part, is passively permeable to ions and impermeable to water. Permeable to water, impermeable to water. And then the thick ascending limb, sodium chloride and potassium are actively reabsorbed, and it is also impermeable to water. So look, the descending limb, permeable to water, the ascending limb, impermeable to water. What does all that do? Let's see what that does. Here. So on the left is a summary of what I've just said in a diagram. So look, here is the descending limb, and here is the ascending limb. Okay, this is the key part. In the descending limb, water is passively permeable to water, so water passes out of the tubule into the medulla. Sodium is passively permeable and goes into the loop here. As it goes around, there is active transport of sodium and chloride, and sodium and chloride is being pumped out. So look what's happening here. If we follow sodium, let's just say sodium, sodium is actively pumped out here and diffuses in here. Goes around, pumped out, diffuses in. Pumped out, diffuse. Sodium is going round and round here. Not much is leaving. Sodium is going round and round. Plus, mustn't forget this arrow here, there's more sodium chloride coming in from the filtrate. So the sodium chloride coming in from the filtrate, and the sodium chloride is going round and round and round. And so not much sodium is leaving the medulla. And if we look at the osmolarity of what's going on in the tubule and in the medulla, you see this pink again means the background here is concentrated. Look here, 300, remember 300, about right. Goes down to 600, 800, 1,000, 1,200, 1,400. This is the same in the tubule as outside, and then back up again when the sodium is pumped out, 12,000, 8, 6, 4, and it comes out about 200. Do you remember I talked about that word net last time? There's little net difference going on in the loop of Henle. Well, here it is, little net difference. 300 going in, 200 coming out. You wouldn't think there's much difference there, and there isn't. It's what's going on in between that's important for our purposes right now. What's going on in between is that as it goes down, it gets very concentrated, and as it comes up, it gets less concentrated. And the important result of this is that sodium and chloride accumulate in the medulla. And it's what's going on in the medulla that's important here, not what's going on in the tubule, because tubule going in 300, tubule coming out 200, who cares? It's the result of all that is that sodium and chloride is concentrated in the medulla of the kidney. Okay, hang in. So as sodium and chloride are transported out of the ascending limb, they diffuse into the descending limb. Sodium and chloride don't leave the medulla in large amounts, obviously a little bit does, and the concentration therefore rises. And new sodium and chlorides continually coming in from the new filtrate. That's the purpose of these long loops of Henle. It doesn't happen in the cortical nephrons because they don't go down into the medulla. We're only talking about the ones that go way down into the medulla that cause this gradient of concentration in the medulla. Now, are there any questions on that? So the purpose of these long loops of Henle is to keep sodium and chloride in the interstitial fluid of the medulla. And it's the interstitial fluid we're interested in here, not what's in the tubule at this point.
So I was just wondering why the osmolarity doesn't continually increase it's at 1,400 now, but why wouldn't it go up to 2,000?
It has to do with the fact that there's water coming out as well. There's water coming out as well, and so the whole thing reaches an equilibrium because there's a certain amount of water there, a certain amount is carried away by the vasorector. I haven't told you about that yet. And the whole thing reaches an equilibrium, and this is the equilibrium. Now, interesting you should say that, because the next two pictures on your handout, this and this, shows you how that equilibrium is reached. And I don't want to go through these in detail today. These are here really for your benefit to have a look at home when you're working through this. What these next two pictures show you are a hypothetical situation where there is no gradient. So here, look, everything is 300. And then you start the pump, you start the movie, as it were, and you start letting in more fluid from the proximal tubule. You start these pumps up, and gradually you see you're building up. Now you've got 300, 400, 400, 200, 200 out, and you play the movie a bit more, and it goes like this, and you let a bit more in here at 300, and now you've got a gradient of 500, and then back up to 150, 200, whatever, and you let a bit more in, and gradually you build up this gradient. This is a hypothetical situation, until you've got about 12, 1400 at the bottom, and an equilibrium is reached. So work through those two pictures and see if you can figure out how the gradient is reached and the equilibrium. And it's not only what's going on in the loop of Henle, but it's also what's going on in the vasa recta. So this picture now is the blood vessels surrounding the loop of Henle. So the issue about those blood vessels are, remember I said it's a special region of the peritubular capillaries. The blood flow is very low, so you're not washing out lots of sodium chloride. You want to keep sodium chloride in the gradient. So the blood flow is low in those blood vessels, so you're not washing out and carrying away lots of sodium chloride. There's also a countercurrent mechanism going on in the vasa recta. Look. See this is not the tubule. This is the blood vessels. And so here's a countercurrent again. Here's the blood coming in and the blood going out. Sodium is pumped out here, and water diffuses in here. Diffuses out, and water in. Sodium out, and water in. And you can also see that there's another molecule important here. It also shows urea. Do you remember when I was talking about clearance? I said we only pee out about half of the urea in the blood, and we keep about half. And the reason we keep about half is because it contributes to this gradient in the medulla. So the medulla is rich in sodium and chloride, but also a lot of urea. And that's because of this this cycle going on in the blood vessels. So look, this is what it says. Sodium chloride and the urea diffuse out of the ascending limb of the vasa recta. Water diffuses out of the descending limb and into the ascending limb. So this reinforces the countercurrent going on in the tubules, and large amounts of sodium would be lost without this going on in the vasa recta. So you've got the pumping going on in the tubules, you've got a reinforcing cycle going on in the blood vessels. It all conspires to give you a high concentration of sodium, chloride, and urea in the medulla. Can I just pause there for a minute? I know that's a lot to take in. The issue that we're addressing is how does the kidney build up a gradient of sodium and chloride and urea deep in the medulla, and a gradient from dilute in the cortex to concentrated in the medulla. And it does it by this combination of pumping out and diffusion of sodium and chloride in the tubules, and sodium and chloride and water and urea in the blood vessels. It keeps the sodium and chloride and urea in the deep parts of the medulla. Okay? All right. Now, why is he banging on about this? I'm banging on about this because this is the key to how we produce a concentrated urine. Next point. Now, this class, we're already at 22. I'm going to tell you how we do it, and then I'm going to continue on next time. So this topic is going to stretch over into Friday's class as well. So I haven't finished with you on this yet. It has to do with the production also of this hormone called antidiuretic hormone. Now, you'll get all this when you do endocrinology, but I need to tell you where antidiuretic hormone comes from. It comes from the pituitary gland. It comes specifically from the posterior pituitary gland here. It's made in the brain. You'll do this in endocrinology. It's made in the brain, passed down nerves, and released here into the blood, into the systemic circulation from the pituitary gland. Here it is, ADH, sometimes called vasopressin. In this diagram, it was called vasopressin. I put the ADH in because that's what we call it here. It comes very close to another hormone, which we're not going to talk about today, called oxytocin. They are very close together, ADH and oxytocin. Don't worry about oxytocin. You can worry about that in a few months. There, look. Did you see that? All about ADH. Okay. So I'm going to tell you here what it is that ADH, how ADH works with that gradient to produce different concentrations of urine, and then we'll go into it again in the next class. If you think about this tonight and tomorrow, we'll come back and we'll talk about it again in the next class and hopefully make it even clearer. I know this looks complicated. It's something for you to look at in your own time. Here we go. Now, here is the cortex. Here is the medulla. Look, it's pale pink here, dark pink here. That indicates there's a gradient in the medulla. We know how this gradient is made. It's made by what's going on in the loop of Henle. There's a gradient in the extracellular cells here. Here is our nephron, proximal convoluted tubule, 300, goes down the loop, 400, 600, 800, 1200, back up out of the loop to 400, 200, 100, 100 here, various things go on here. Various things go on here with potassium, we know about that, there's the potassium loop. Sodium coming in, and then it dives back down through the medulla. What are the tubules seeing as it dives back down through the medulla? The tubules are seeing a very high concentration of sodium chloride and urea out here. Now, if these membranes are permeable to water, what's going to happen to the water in the tubules? If these membranes are permeable to water, what is going to happen to the water in these tubules when the tubules dive back down through this high concentration of sodium chloride? Remember osmosis? Hypotonic, hypertonic? What's going to happen to the water? It's going to come out. So the water is going to, if these channels are open here, as the fluid comes down here, water is going to come out and osmotically in response to this high gradient. And what you're going to end up doing is, if there's lots of water channels here, aquaporins, your P is going to come out at the same concentration as the concentration of the gradient. Right? What happens if there aren't any aquaporins or water channels in these membranes here? If there aren't any water channels in there, water can't come out. And so, if there are absolutely no water channels here, water can't come out. Doesn't matter about the gradient. There's no way for the water to get out. Then you're going to pee out unmodified filtrate, and you'll see you're going to pee out 300, 400 milliosmoles of pee. So it all depends on whether you've got these water channels going on here. Put a ring around that. A ring around that. Because that is what determines whether or not you pee out concentrated or dilute urine. If you've got water channels here, then the tubules respond osmotically, or tubule fluid responds osmotically to the gradient, and you pee out concentrated urine. If you haven't got water channels here, the filtrate in the tubule can't see the gradient, and you pee out dilute fluid, because water can't be reabsorbed. Get it? So your question is, what determines whether or not there are water channels in these membranes? The answer is ADH. If you have ADH, you have water channels, and you pee out small amounts of concentrated urine. ADH, antidiuretic hormone. You don't pee much, and it's very concentrated, because ADH puts water channels in these membranes. If you don't have ADH circulating, you don't have water channels in these membranes, you pee out large quantities of dilute urine. Get it? If you don't get it, we'll come back and we'll continue with it next time. Hope you get it. Okay.